my honor to argue professor. Yeah, Professor oron catt from University of Western Australia. And his official title is Director of the Institute of Advanced Studies, like IAS, at the university. But at his spare time, he's also artist and creator, and very interesting artwork. He's been very senior and pioneering in the field of bio art, and he was the first artist to make art using raw meat. Okay, so today we're very excited to hear his lecture about interdisciplinary lessons from symbiotica. Okay, so what a cool name. All right, so now the floor is yours.
Thank you so much. Yeah. It's so great to be here, and thank you so much for the invitation. The talk I'm going to give you is kind of a bit eclectic because I want to take you through my journey. So I'll start from the end, but then I'll jump into how I started, and I'll talk about some of the art and science initiatives that I was involved with, and I'll talk about my artwork as well. But I suppose in order to start with, we have to contextualize it. This is a quote from the World Economic Forum that talks about the fact that we live in a really interesting time, and I think we all agree, and especially here in China, I think it's like one of the most interesting times, at least in recent history, in regard to the fact that, and I think China is a really great example, that we're here in a place where the 21st century science is taking place. And actually here the technologies, I would say, are already 21st century technologies, but it's still very much in 20th century mindsets, and to a large extent, even an institution like this one is still very much based on a 19th century institution. So there's a kind of misconnect between how our institutions and we talk also about political systems, everything seems to be a mismatch in regard to the kind of knowledge that is being generated. And again, here is like one of the most advanced places of production of knowledge, but you still have to deal with the fact that the knowledge is almost incompatible with the way society receives it. And as an artist, I'm really much more interested in the mindset than the institutions or the technologies, but we need to figure out a way to bring those things together. So the way to get culture along this journey, it's not enough to tell them and from top down how to do things. It's about kind of getting them engaged in a cultural way with what's going on, but also having a voice into how the knowledge, because there's a difference between how we find out things about the world and then what we choose to do with the knowledge that we have. And this choice of what we're doing with this knowledge shouldn't be just left to scientists or engineers or companies or governments. It's all of us together. Figure it out.
Yes, please. So the fact about this room, and the audience in this room, I think echoes perfectly to this quote.
Yeah.
Right. So now you are sitting, on a top-notch institute of cutting-edge science.
Yeah.
But obviously, most of the scientists basically ignored, right? And we had at the same time people coming, half of China from the central China.
Yeah.
To visit you. So I think that's precisely the problem actually we're trying to tackle.
And how you open it up. So
exactly.
That's,
Yeah.
So thanks. I'm more than happy to actually, because we're a small group, we can read the conversation. So my current job, as you mentioned, I'm the director of the Institute of Advanced Studies. Now, Institute of Advanced Studies have a long history. The very first Institute of Advanced Studies was set up in Princeton, in America in the 1920s. The director of the founding director wanted to set up what he talked about as a monastery for scholars and researchers. One of his very first fellows was Einstein. He brought Einstein from Europe. It was also about when you think about kind of the way the world was in the 1920s and 1930s, there was a major political shift. So he was trying to save scientists from increasingly fascist Europe and bring them to America so they can engage in research without any external pressure. And that was one of the things. It's like bring scientists, give them everything they need. They don't have to apply for funding. They talked about it as the researchers' paradise. My institute I wish it was like that, but we are trying to do something similar, especially around supporting interdisciplinary research. So, as I'll show you later, my main interest was always art and biology, but with my current job, it's interdisciplinary research across the whole disciplines of the university. And our university is a very general university, so there's business schools, there's geography, geology, obviously mining and engineering, English, radio, art, and cultural studies. So I try to bring people together through bringing international scholars to my university. So this is just to put you in context into what I'm doing now, but I'm still continuing with my art project. This is an example of one of the recent artworks that I've done. And the reason I brought it here because it's kind of a similar situation to where I am. So this is from last year, and to what's happening now here. So I was brought for a 2-week residency at the School of Molecular Medicine at the University of Lisbon in Portugal. And they wrote me and said, just come and play, but then, by the way, we want you to produce an artwork. As an artist, it takes me about 3 years to produce work, and that was 2 weeks. Make an artwork now, because we have an international conference of immunologists, and we want you to produce something for it. So I did a small piece, and it seemed to be actually quite successful. Since that showing it for the first time in Ireland, it's a meeting of the immunologists already been exhibited in South Africa, and here in China, and in a few other places around the world. What I've done with this was to take a mouse immune cell, put it under a microscope, and heat it up to 45 degrees Celsius. Basically, give it fever, and then the way, and made a time-lapse video of that, but the way we present it is projected on a dome, and it looks like a planet. And once in a while you would see... but basically you would see, we superimpose it on the globe, so soon you might be able to see actually Western Australia kind of popping up in the corner there. And the idea was to compare, or to talk about the idea that global warming is like a planetary fever. So the same way that our immune system responds to infection and increases the body temperature, we can also say that human activity at the moment is almost like a disease that gives fever to the planet. It's a very simple story, but the way we project it here, you can see Western Australia. Yeah, so you can see the planet. Yeah. And people kind of tend to get it. And obviously what you actually see is what happened to the cell as it's getting hot, and in some stage, obviously the cell died. Yeah? And so kind of a very simple 2-minute time-lapse kind of loop, but quite an effective kind of narrative that seems to people tend to like it. And obviously it's a really easy piece to show as compared to many other works that I find that are much more complicated. Now, why it is important for artists to work with science, this is one of the best examples I have from the early 20th century. This is the first incubator award or premature birth award in the United States, 1905, in Buffalo in upstate New York, which was part of this was set up as part of the Pan American Expo. And so you can see the babies inside those incubators. Those systems had about 80% success rate. Those premature babies would die if they wouldn't be put in the incubator. And if they were treated well, 80% of them survived in those conditions. But you can see how there's like Disney World style crowd control rails there. That's because it was basically considered to be a display of oddities. And it was, the interesting thing about the Pan American Expo was it was supposed to be a celebration of the new America, kind of the turn of the 20th century, but that wasn't in the technology section. That was in the kind of weird stuff section. And we know that was there because when the Pan American Expo closed down, everything moved, all of this installation moved to Coney Island, to the freak show. And you can see there it says infant incubators with living infants. So people would go and pay tickets for a ticket to go and see those babies in incubators because it was so strange to see human bodies inside machines. Now, there are many reasons as to why hospitals didn't want to deal with incubators at the time, but from my perspective as an artist, I can tell you that one reason is because there was no cultural place to put those things. Yeah? So, as a culture, as a society, it's like, where do babies or human bodies in machines belong? And the only place they could put them was as freaks, in the freak show. Yeah? And so it is important because this is exactly the situation we find ourselves with new biological knowledge. If we don't have a way to culturally articulate it and discuss it and engage with it, it would end up as freaks. This actually lasted for about 40 years. And even when hospitals started to use baby incubators, it was sold from Dreamland, Coney Island to both hospitals and amusement parks. Yeah, so this kind of weird place where the in-between spaces, and again, that's where me and my artist colleagues who are working with biology usually find ourselves. Maybe another example would be even illustrating better. This is Dolly the sheep. So Dolly was the first animal to be cloned back in 1996. It was a huge technological feat. Yeah, it was like one of the biggest technological breakthroughs of the late 20th century. When Dolly died, the Rosalind Institute, where it was, where she lived, donated the body to the National Museum of Scotland in Edinburgh. And the people in the museum were like, where does Dolly belongs? Is natural history or human artifact? What is Dolly? Where does she put? So obviously, in order to prepare the specimen, they had to prepare it in the natural history section of the museum. But then the natural history curator said, it's not part of, we put wild animals, it's not part of us. And this is kind of, I really like this photo because this is Dolly being moved from being prepared in the natural history section of the museum to where it ended up being displayed, and that's in the human artifact, human culture part of the museum. Yeah, so this is kind of where I put myself. I'm exactly there in the corridor between life and technology, yeah, between culture and science. And we can think about it also in those terms. There's some really strange things about how we treat those new form of technological artifacts, which are also living biological things. I was really surprised. I went to the Science Museum in London, and I saw this. You go to many museums, many museums want to have a piece of Dolly, so you might find a museum that would have a sweater that was knitted from the wool of Dolly, but the Science Museum in London, the curator there, actually it was the curator of the Wellcome collection, decided to collect Dolly's droppings and preserve them. So you can go to a museum and you can actually see this technological shit, literally. Yeah. And this is something which is accepted in a sense, but again, this is kind of how we move from the freak show to the understanding of technology. But I can tell you, and this is again, me as an artist, I would have been much more interested in having on display one of the 277 failed attempts. Because we also have to understand that science is a trust. And there's ethical issues that we need to consider. But I can guarantee there's no museum, and obviously the research institute would never release a sample of the failed attempt to be shown in public. But how we can create a kind of a broader conversation around it, I would love to put my hands on one of those. So my background, I actually studied product design back in the 90s, in the early 90s, and I was also interested in biology. And as I was studying, I was kind of thinking what would happen if we would start, as biotechnology was progressing, so late 80s, the Human Genome Project started. There was quite a lot of discussions around how those new biological technologies are going to impact, and the move of biology from being just a descriptive science where we describe the world around us to a prescriptive science, basically to engineering. Yeah? So we moved from trying to understand how life works, to trying to understand how we can use life for our own purposes. Yeah, so this shift from science to engineering. And I was speculating what will happen once the engineers are starting to engineer life in different ways. Would they start to call designers to design living biological products? So I wrote what, at the time the term didn't even exist, but I wrote a speculative design thesis about the future where design would be part of biology, or designers would start to design living biological products. And I deliberately chose skin or surfaces, and how kind of if what happens if we cover, that was my thesis, what happens if we start to cover domestic objects with living surfaces? How are we going to relate to them? Because we as humans, we obviously interact mostly with surface of things. And so if things would start to appear as if they're alive and they would have some living components, would our relationship to the objects around us be different? So you can think about the difference between how you treat a piece of furniture and how you treat a plant. Yeah, there's a very different. We treat it and think about it differently. At the time, I was very enthusiastic about it because I thought this would be a great thing. Yeah, because first of all, if we work with living biological materials, they're much more compatible with the environment, and therefore they'll be much more environmentally friendly. We would move away from just treating our objects as something that we use and throw out to something that we care for. But as I was thinking about it more, I realized that there's also a flip side, because if we start to think about life as something that we are, as a technology, it might impact the way we relate to life in general. Yeah, as a commodity, as something that for us to exploit and extract value rather than care for it. And the other thing, which is retrospect, is quite obvious, not all living biological solutions are actually environmentally friendly. Yeah, think about the issues we have with industrial farming. Yeah, so that was a solution to mass production of meat, and it's one of the worst environmental ways of dealing with food production. So the fact that things are biological doesn't make them environmentally friendly necessarily. Yeah, so we need to think about those issues. Yeah, so I got much more interested in the questions that this kind of approach is raising rather than the solutions. So I moved from being a designer, and design by definition is an optimistic profession. I'm a pessimist, so I moved from design to art to be able to continue asking questions. Art is a very privileged place where you can engage in open-ended questions. You don't have to come up with solutions. You raise questions and you challenge the perceptions rather than closing things down. Yeah. So it's really about opening things up. And the technology that I was really interested in was what was called tissue engineering, and it's now known as regenerative medicine. And it is kind of a really interesting conceptual shift in how we were thinking about life. I was very fortunate to spend a year in Harvard Medical School with the scientists that actually developed the whole field of tissue engineering, Professor Joseph Vacanti, who wrote the original tissue engineering paper back in the late 80s. And those slides are actually from the presentation that he gave to the third International Tissue Engineering meeting, which, funny enough, he organized in Disney World, which is, again, the idea of the freak and the sideshow. He deliberately wanted the meeting to take place in Disney World. He was in the group of scientists that made a very famous mouse that had a human ear on its back. And we would sit down, I was sitting at the same table with him for breakfast in the Disney Hotel, and we would be served breakfast by Mickey Mouse talking about mice with human ears. Yeah. So it was kind of a very surreal moment. But this is from the presentation he gave to the meeting. We was very proud to show how the idea of repairing bodies changed. So this image over here is from 1989. At the time, the idea was that if a body part is failing, if we need to replace, if we're missing a body part, we would replace it with the dry and hard product of electrical and mechanical engineering. And so this idea that bit by bit we would replace our bodies with literal machines. And that doesn't work. The body is corrosive, the body obviously is self-repairing, those kind of machines might work as a temporary solution, but for really repairing and fixing human bodies, this is not the optimal solution, even though now there's hybrid ways of thinking about it. And then exactly 10 years later, in 1999, the whole story changed. Suddenly, the body becomes its own technology of repair. Biology is becoming a technology of repairing bodies, and they imagine that somehow they'll be able to harness those latent regenerative powers of our body. They're saying, okay, if a salamander can regrow a limb, why not humans? Yeah, so, and we can somehow find the switch that would allow us to regenerate. And the solution that they were thinking about then, which was tissue engineering, was the idea of creating a three-dimensional scaffold out of a special degradable polymer that is bio-friendly. And so let's say you want to replace an ear, for example, so you basically sculpt the shape of the organ you're trying to replace using this polymer. You then take cells that you harvested from the patient itself, you grow them outside of the body onto this polymer structure, and this is where the art comes in, because it's very much like a lost wax system when you do jewelry, for example. So artists really get it, and this is a very obvious way of creating new forms out of and replacing materials, in a sense. And so you then put the cells, they grow, they start to replace the polymer, and then you implant it into the body, and you're supposed to have a new body part that is functioning, it's biological, and it's grown out of the cells of the patient. I can tell you this is not how it works. Yeah, it's, that was since the early 90s, scientists were trying to do it. The only clinical work, clinical solutions we have now using this technology are very, very simple organs like a bladder or skin. Yeah? Making complex organs using this technology is almost impossible. But as an artist, again, I was like, this is quite an amazing technology to be able to start to grow sculptures. And also, actually, even thinking like a designer, I was thinking, so if in the 1990s we were told, actually there was an international consortium of scientists that claimed that in 10 years' time, so from 1995 to 2005, we would have fully functioning human hearts that are grown in such a way. And then in the 90s we were actually told that we can replace the hearts with pumps. So I was kind of reversing this logic, and I said, if this technology actually works, why not grow pumps? Yeah. And so I was interested in the idea that, forget about putting it back into the body, can we grow three-dimensional functional products outside of the body and never put them into the body and start to use them? But as I said, I was much more interested in the question that it raises. So in 1996, I set up the Tissue Culture and Art Project exactly to try and deal with those questions and ask even more questions. And I'll talk more about the Tissue Culture and Art Project shortly, but as a result of setting up the Tissue Culture and Art Project, which one of the things which was really important for me was, like Feynman was talking about, if I can't do it, I won't understand it. Yeah. I wanted to understand it myself by actually doing it. So I was very lucky, I was able to get into a lab and start to work in the lab and do tissue engineering myself as an artist. And this model of hands-on learning of the techniques became then a model for a lab that I set out together with a couple of scientists in the year 2000, and that was Symbiotica. So Symbiotica was the first level 2 biological lab specifically dedicated for artistic research. And we were part of a science school, so we were part of the School of Anatomy and Human Biology at the University of Western Australia, but we were doing artistic research rather than scientific research. And this is kind of the lab that we had there, like, looks like any other lab, but in this case, there were a bunch of artists experimenting and doing work. So we were set up, okay, there's an issue with the font there, but anyway. We were set up as an artistic research laboratory dedicated to the research, learning, critique, and hands-on engagement with the life sciences. we strive to articulate and provide new cultural language to the shifting relations and perceptions of life using art. So the idea was very much around kind of dealing with this gap, with the growing gap between how we as a culture and society were thinking about life and what was going on with life in research institutes. So our research scope broadly was around exploring those philosophical questions around those shifting perceptions to life. We would spend a lot of time reading scientific literature to identify the areas that we believe need more cultural attention, but also areas that gives us a potential, because as biology became more and more of an engineering pursuit, it opened up a new palette for artistic possibilities. One of the things that engineers are doing really well is de-skilling, so creating protocols and systems where, if you think about how people who did molecular biology, 30 years ago, you needed to have a PhD just to do simple genetic engineering. Now, primary school kids can put GFP in bacteria quite easily. So there's the whole kind of the building up of those protocols creates the ability and opens up possibilities for artists to engage with life in many different ways. We were then also very interested in what it means to move those things that are happening in the lab and putting them within a cultural context and into museums and galleries. Now I can tell you from my experience, and it's still going on, museums and galleries, those are those 19th century institutions. They're designed against life. And I'll be provocative and I would say those are places of death. They are designed to keep dead things as dead as possible for as long as possible. That's their purpose. What can you do about it? And what happens when an artist comes in and wants to show something which is living? It's a struggle. So we had to develop all sorts of different strategies of how you work with museums and galleries, how you explain it to them, how you display this kind of work, and how you put it within a cultural context without too much issues. And we constantly had, we develop protocols, obviously things around health and safety, but also more conceptual and philosophical approaches to dealing with that. And sometimes we would have to develop our own technologies as well. And here you can see in the background, this is a sketch that we had a designer in resident, Matt Johnson from the Royal College of Arts in London, who worked with an artist called Alicia King. She was very interested in showing living tissue, like many of us, and they worked together on designing an instrument, a bioreactor, to her specifications. Now, if we would try to get a piece of scientific equipment to do it, it would cost in the thousands, if not tens of thousands of dollars to do what she wanted it to do, and it would be opaque. It would be, you won't be able to see it. Yeah. So they worked together and they developed this device, which you can see here in the background, where she was able to show and keep living tissue for a fairly long duration of time in a gallery situation. You can see very kind of DIY style with this fork. Actually, that was illegal. We were showing it in the EU. We had to import this from Eastern Europe, the heating element, because it was illegal to buy in Western Europe. And we had to cut quite a few corners, and it cost us about $500 to do it, to the specification of the artist. So this is one example of many where we developed our own technologies, both because we needed them, but also because we couldn't afford the scientific equipment. Now, when you think about it, scientific equipment is built for high precision, and the more precise it is, the more expensive it gets. You reach a level where every 0.1% of precision increases the cost of the equipment tenfold. Yeah. So because we didn't, as artists, we don't really need to engage too much with the idea of reproduction or reproducibility of our experiments. The precision was less important. The robustness and the ability to design it to our purposes became more important for us, and obviously allowed us to do things which were much cheaper. So in Semiotica we developed academic courses. We were the first place in the world that offered a Master's of Biological Arts, which was actually a science degree because we were part of the science department. And we ran exhibitions. The main thing we've done was artist residencies, and then different kind of dissemination of the knowledge through symposia and seminars and I would run those very intensive workshops teaching artists, and I ended up finding out it wasn't just artists who were interested with some basic biotechnology techniques. So it would be a week-long workshop where we would do everything from basic genetic engineering to plant and animal tissue culture, but that would always be accompanied with conversations about what it means to do those things. Yeah, so it wasn't a science course, it was very much more art and philosophy, but by direct hands-on engagement. This is a partial taxonomy of the areas that we engage with. Another thing we found out, even though it was supposed to be for artists, quite quickly we started to get people who, social scientists, SDS people, so Science Technology Society people, geographers, ethicists, philosophers, obviously designers, architects, people who were interested in knowing what's going on with current biological research, but never got an opportunity to get into the lab. So we became the conduit to do it. And then we started to get applications from scientists who wanted to do a residency with us. Some scientists, even that had their own labs, realized the only place that they can engage in what they were actually interested in was in our lab, because they were so limited by funding structures and what they were told that they need to research, that they would take a sabbatical and come and do research into the areas that they were interested in with us. It was kind of hard to convince funding bodies. What does it mean to get funding for a scientist to come into an art lab within a science department to do kind of crazy open-ended research? But we had some really interesting things going on there. But there's issues. So obviously there's an issue that there's still, and this is something that happens a lot, there's still a perception that artists are like this person here on the left, yeah. A painter, someone who's doing work which is representing rather than presenting the issues. So we would often get scientists thinking that the only purpose the artist would have is to make beautiful pictures or illustrate the science, and they would get really confused when they would see artists actually working in the lab on the bench with the living biological material themselves. Yeah, so how you break those barriers. I realized that in interdisciplinarity, it's almost like to migrate to a new country. And I did find out that people who were migrants had a better understanding of working between disciplines because they already went through this experience of moving between cultures. And science is a culture. Yeah, so how you communicate and how you come, there's something quite strongly there. There's also the issue in regard to how science and art are different in the way they're being credited, but also how the media credits those types of work. So this is a recent work by some of my colleagues that developed the project at Symbiotica, a very complex neuroscience project where they use brain organoids from a deceased composer to compose music in a gallery. And this is kind of a headline in one of the newspapers that a team led by a neuroscience. And neuroscience is the guy on the left, the bald guy, but actually the person who led the project was an artist guy, the guy with the beard over there. Yeah, but the media couldn't understand that a complex neuroscience project can actually be led by an artist, where the neuroscience was actually just working with them. Yeah, so, but then there's the other issue that we are slowly getting recognition for the idea that artists can work as groups, as teams, not just as the genius solo artist, because there's still a perception that there's like an artist is this guy that works by themselves, and also the way, especially around the commercial art world, it makes sense. You want to put one name so you can then put value to this name in the art market. But we're doing experimental art, it doesn't really important for us. And then within the sciences, obviously, almost everyone who's involved in a project is being credited as an author on a scientific paper. So how you manage the tension between crediting is still kind of an ongoing issue that we're facing. I must say that I ended up being the sole director of Symbiotica because the main scientific director of my lab of Symbiotica kept on asking artists to put his name on their artwork, even though he would do nothing. But he said, I'm the head of the lab, like this is common, you know. And the artist would say, that's not how it works here. And it became such a problem that the university decided to remove the scientist because it said, this is not how it should happen. Then there's things around language. The very first time we showed our live tissue engineered sculptures in a gallery, we asked the architect that designed the display to set up a display system that would keep everything in 37 degrees. And for us it was obvious, it was 37 degrees Celsius. But when we arrived to the show, the architect said, I didn't build it because I didn't understand why you wanted the 37 degrees angle and why are you so specific about it. Yeah. So even those kind of things that seem to be obvious in one discipline are very different in other disciplines. So being sure that you can somehow cross those barriers and understand what you're talking about is something that is quite still an issue. And then there's what I refer to as the integrity of discipline. So for me, even though semiotica was a very interdisciplinary research lab, it was important for me that people would know where they are and what role they're playing in those collaborations, in those projects. The worst thing that happened to us was when scientists decided that they're artists and when artists decided that they're scientists. Because then artists are not trained as being scientists and they don't, in most cases, they won't do any good science, and it's the same with scientists. Yeah? We had cases where a scientist would work with an artist on a couple of projects and then it's like, no, I want to be the artist now. And it could have been quite a problem, but also the way a project is being read, if it comes from a scientist, the audience would read it in one way. If it comes from an artist, they would read it from another way. So the issue around the integrity of disciplines is something which have to be kept in those interdisciplinary relationships. So when you think about, I like this metaphor, so there's, we know about transdisciplinary, interdisciplinary, cross-disciplinary, antidisciplinary, all of those terms are being thrown around, and there's very few kind of definitions that actually make sense. One thing I read which makes some kind of sense is it's like cooking. Yeah. So transdisciplinary is like baking, where you take all of those ingredients, the different ingredients, you mix them together, and something new comes out, and you can't see the defined original ingredients in them. Interdisciplinary, which is where I see myself, is more like curry. Yeah. You still see the bits of the ingredients there and you know how to identify them, but they're creating something new and new flavors, but there's still kind of a definition and you can actually separate them almost to the original state. Right, so now I'll talk a little bit about some of my projects. As I mentioned, the very first, back in the year 2000, I was able for the very first time to show live tissue engineered sculptures in a gallery. And I decided, together with my collaborator Linat Zuro, to do our own version of the Guatemalan worry dolls. So the Guatemalan worry dolls are basically small dolls that indigenous Guatemalans would give to kids. And the idea is that kids can tell the worries to the dolls, put the dolls under the pillow, and the dolls were supposed to take the worries away. And for us it was important that the very first tissue engineered sculptures that are shown are kind of dealing with issues around anxieties. And actually the kind of one of the triggers to do this piece was when, so as I said, Yonat and I were lucky enough to be invited to work in the lab of Joseph Acanti in Harvard. And when he saw us under the hood, even though we had him, he invited us, he still in his mind thought that we would be like the artists that would paint. And then he saw us under the hood, because by that time we already had 4-year experience of working in tissue engineering, and he was quite shocked. And I said, okay, I'll borrow you a worry doll, and you can whisper your worries to the dolls, and that would take the worries away, and we would be allowed to continue our work. And luckily for us, he had a sense of humor and said, okay, that's good enough. So we did grow one worry doll for him, and it's like, okay, if you're concerned about what we're doing, just whisper it to the doll. Now, with worry dolls as well, the Guatemalan legend talks about the fact that kids are only allowed to have 6 worry dolls because kids are only allowed to have 6 worries. We did 7, yeah, because we're not kids anymore. And so we were invited to Ars Electronica, which is one of the biggest art and technology festivals, and because it was the very first time anyone showed tissue engineered sculptures alive in a gallery, we were trying to figure out how to do it, and we decided to set up a fully functioning tissue engineering lab in the gallery itself in this kind of clear tent. And that was a mistake, I can tell you now, because most people didn't see the worry dolls. all we saw was a bunch of artists taking scientific equipment and putting it in a gallery. Or in this case, it was the foyer of the local opera house in Linz. And the warriors were tiny, yeah, so they, so this is kind of a great example of what happens when artists are working with new technologies, that they always run the risk that the technological frame would take over the content. Yeah, so how do you create an artistic story, a narrative with new technologies without the technology taking over the story? Yeah, so that was a really good lesson for us. Those were kind of the dolls, the before and after. We used very similar techniques to how the Guatemalans would do it, but in our case, we used degradable polymer and surgical sutures, while the original Guatemalan dolls are made out of fabric and string and thread. And we then seeded them with cells and we grew them in those microgravity bioreactor. Now, we were trying to also show to the audience that those things are alive. So we would do feedings, basically feeding sessions like the feeding times in the zoo. Because we needed to feed those cells once every couple of days, but we decided every day during the festival to go into the lab and basically perform the feeding ritual, which was something that those who work in the lab know. It's a really boring piece of work where you just suck out the old nutrients and you put the new nutrients there, but that was kind of a way to try and present that those things are alive. We also had a way for people to tell the worries to the dolls. We were trying to push them to see what kind of anxieties they have in regard to those new technologies, only to find out that most people gave those dolls almost magical powers and were expressing the most intimate worries and concerns rather than what we were trying to do. But that became a really interesting database of people's anxieties, because we showed this work for almost 20 years in different configurations. And then in the year 2000, we were invited to present work that was supposed to celebrate the so-called completion of the first draft of the Human Genome Project. So those of you who are old enough might remember June 2000, there was a big ceremony where Bill Clinton and the head of the NIH and then Tony Blair and the head of the Wellcome Trust and whatever the British Medical Research Council is, had this celebration where and Craig Venture was there as well, because
he's one of the competitors.
Yeah, exactly. And the reason for that event was that as Craig Venture, which is a private company, started to take over the public
credits before the public effort.
And it created a big political stir, because the Human Genome Project was one of the most expensive public science projects, and suddenly this private company taking over and doing it much faster, but then also patenting the genes as they're finding them. And so they decided to declare the Human Genome Project complete, even though it wasn't complete then, just as a way to get the media. It was a PR exercise rather than. And the rhetoric around the Human Genome Project was amazing. Yeah, so they would say, they did say actually in the ceremony, now when the Human Genome Project is complete, the impossible is possible. And in English, and I think in Chinese, there's also a similar term that once pigs would fly, everything would happen. Yeah? So everything is possible. Yeah? So we said, we picked up on that, and we decided to, and said, okay, so you're basically saying that pigs can fly now. Yeah? So we wanted to see what kind of wings those pigs are going to have. So we looked at the three evolutionary solution of flight invertebrates, the birds, the bats, and the dinosaurs, and we used bone marrow stem cells from pigs. Actually, it was also funny enough. We introduced 3D printing or 3D modeling and 3D printing to the Harvard lab, but rather than growing an organ, we use it to grow those wing-shaped objects out of pig tissue. I designed on the CAT program those three sets of wings. We then printed them, made them out of this polymer, and then we grew them in those microgravity bioreactors for about 9 months. Until, because we're using bone marrow stem cells, we then differentiated them into bone, and we ended up with those tiny bony structures in shapes of those 3 sets of wings. Actually, funny enough, the bat wing over there in the decided to grow its own set of extra wings on top of it as well, which is another thing, is the control over how those tissue is growing is not complete. Yeah, there's always kind of biology fights back and comes up.
So you have to use microgravity more than otherwise they will grow evenly, right?
And also for the thickness. So this one of the things microgravity system created was also mass transit of nutrients through it, because obviously, and this is still an issue, there's no capillary system.
Yeah.
So even bone, what we're trying to do, that didn't really need a lot of nutrients. You still have to wash the nutrients through it. So at the time, there were some of the biggest three-dimensional tissue constructs ever to be grown. Obviously, there's the thing like the mouse we did, or the ear on the back of the mouse, but then they used the mouse, and the mouse kind of migrated blood vessels into the construct. But as detached constructs, which were not attached to the body, they were some of the biggest constructs, and to some extent still now. They're about 4 millimeters thick, about 2 millimeters long, and about kind of 4 millimeters, 2 centimeters by about 1 centimeter. Yeah. So not that big.
No.
Now, conceptually, the idea was that in Western iconography.
So what does the color come from? Just lights.
So the color was, when we done the imaging, we basically developed a stage lighting. So it was almost like with LED lights, and because actually we kind of cleaned it quite a bit, but each one of those images is about 300 microscope frames.
Okay.
Yeah, so, but we wanted that the light would be consistent, because otherwise we did it with another project, so if you just use the light source from the microscope, you would have like this bright area in the middle of the square, but then it would darken off. So we wanted, so basically the LEDs were traveling on the stage with the specimen. But when you think about the iconography of wings, so in Western society, if bird wings are attached to something which is not a bird, they're usually attached to angels. Bat wings are usually attached to devils and demons. And the paradoxal wings are kind of, we still don't know where they are culturally, because we just found out about them late in our history. There's claim that there might be kind of dragon wings, but again, we don't know if dragons are good or bad. So the idea was to look at those 3 ways of thinking about those new technologies, or about in this case, about the Human Genome Project, and say, it can either be angelic and save us, it might be the devils and demons that will destroy us, or it's like, wait a minute, we still have to figure it out. So the body that commissioned us was the Wellcome Trust, actually, which was one of the main funders of the Public Human Genome Project. When we sent them the proposal, they sent us the nastiest but most honest rejection letter an artist can get. They basically said that they were so pissed off with us because we were questioning. They wanted us to celebrate. They wanted us, if we would be critical as artists, maybe critical of Celera, but not critical of the hype. So they basically said that our work didn't have any artistic or scientific merits. Now, at the time, as I said, we developed a protocol for 3D printing of tissue, or 3D printing of scaffolds for tissue engineering at the top tissue engineering lab at Harvard. That wasn't good enough for them as a scientific thing. And artistically, this is now in the permanent collection of MoMA in New York. So sometimes rejections are good, and sometimes actually, you know what I'm saying to my students, if you are going to be rejected, make sure that you're rejected in a way that they would tell you the kind of the true reasoning would come out, because usually rejection letters are very polite. Because the other thing they said, which was really interesting and became part of the project, was that we didn't represent the public opinion in regard to the Human Genome Project. And this is really interesting because artists not supposed to represent public opinion. Artists in society are usually being celebrated for thinking differently than the rest of the world. So they were trying to, basically, they revealed the fact that they wanted us as artists, the way we sent an apology saying, we're sorry that we didn't represent what you think the public opinion should be. Yeah. And for a while it was actually part of the project. We showed the whole exchange of the with them. We ended up showing it, so as I said, now it's in the MoMA collection, but the very first time we showed it was in a big art biennial in Australia. But what we did find out also that art galleries were also hyping up the project. And people would come expecting to see, if not flying pigs, at least wings which are big enough for pigs to fly with, and then would come and would see those tiny objects in those cheap jewelry boxes. And that's when we started to talk about the aesthetics of disappointment. Yeah. So this importance of artists sometimes not to be part of the hype, but actually engage in the disappointment. And as I said, those were some of the biggest tissue engineered constructs, but they were still quite disappointing. So this is one version where we show the live. So again, we set up a lab in the gallery, but a much smaller lab where we show the live version. We also then devised what we call the killing ritual. So what happened is that in many cases we would, those things require a lot of care and maintenance, and often being based in Australia and showing our work internationally, we couldn't stay in the gallery for the whole time of the exhibition. So we would kill the sculptures at the very last day that we would be there, as a way of kind of almost like a form of euthanasia, and saying, because it's our responsibility as artists to look after them, it's also our responsibility to be those who are dealing with the disposal of them or the killing of them. And that became quite a big thing, where we would invite the audience to come and kill our sculptures by touching it. And more than once we got this response that people didn't believe that those things are alive until they killed them. Yeah. So this idea of, again, and you can imagine how nervous museums and galleries were when those things were going on. Talk about this project, because this is another really interesting one. So the Victimus Center is one of our most famous works. We developed it in 2004. In 2018, I was invited to this Biofabricate conference in New York. So Biofabricate is a conference that started around 2015 by a designer called Suzanne Lee, and her claim to fame is that she was the first creative director and an executive of a biotech company. So up to that time, obviously, there was no biotech company was thinking that they need a creative director, and obviously not putting one of them on the executive. But she was invited to be on the executive of a company called Modern Meadow, and as a result, she also decided to kind of open up this whole area of what she called Biofabricate, and that's as basically my thesis from the mid-90s became true, where designers were starting to work with biotech companies to design living biological products and design biomaterials and so forth. And so this this event happened in New York. I think that was the third or the fourth time that they're running it, and they decided to do a timeline of this new approach to designing or using biotech for consumer products. And they made, in the timeline, basically the very, very first thing on the timeline is the Victimus letter from 2004. And basically the trigger that opened up this whole new field. I was both humbled and honored, but also quite disturbed, because that was never my intention. Yeah, it wasn't. I didn't want to start this new technological revolution, and I was kind of scratching my head as to why they chose this one to be the trigger, because we didn't develop any new technology. We were using existing technologies, but what we've done was to, in a sense, liberate the knowledge from the biomedical world and put it in a different context. And by that, basically allowing other people to think about, how can those new technologies can be used for other purposes. And so this interesting thing about, the whole story about gentrification, where artists are moving to a very poor neighborhood, and then people are coming in because suddenly it's sexy, because the artists are there, and then it's becoming too expensive, and the artists are being pushed out, and they have to move to another suburb and do it the same. So in a sense, artists are also doing gentrification to knowledge. Yeah, so what artists can do is make knowledge sexy and open it up, and then other people can come in and use it in other ways. And the victim study was a really interesting example of that, because we did research how to grow leather in the lab. We actually worked with a bunch of science students, we did experiments, we're looking at the best way to grow leather-like material. But when we displayed it, we decided not to display the leather products that we've grown, but to display this thing, which is basically a tiny jacket in this bioreactor that we designed, that is kept alive for the exhibition, and kind of reversing the roles as well. So in a sense, we think about leather as something that protects, but in this case, the leather is the most fragile. It needs to be protected by all of those layers of technology that are keeping it alive. It also was kind of standing for the body, so the bioreactor itself, we have the nutrients, which is like the blood, we have the pump, which is like the heart, we had the gas exchange chamber, which is like the lungs, and created like a minimal technological body where the leather can grow, where this tiny jacket can grow. When we showed it in MoMA in New York in 2004, something quite interesting happened. So in 2008. So when we were using, in this case, so we were experimenting with different types of cells, and in this case we used mouse embryonic stem cells, and they grew so fast that they sheared off, they fell off the polymer structure and started to travel in the system, and at some stage they kind of blocked the tubings. And we got a phone call from the curator, and we said, okay, we need to turn it off. And what was really interesting was that rather than hiding it, the curator decided to go public with it. And you can imagine in America, this conversation around killing a jacket made out of stem cells from embryonic stem cells from a mice, when the whole debate about abortion is taking place, it became quite sensationalist. And basically the media had a field day, yeah. So I never thought that the New York Times would have a headline saying that MoMA kills my art, but that happened. Art Asia Pacific had murder in MoMA, and so forth. Now the exhibition itself was actually a design exhibition. It was called Design and Elastic Mind. The idea of this exhibition was to show this bright future where designers and artists would work with scientists and would find solutions to the most pressing problems of our times. And this was the only living, there are about 250 works in the exhibition. This was the only living work in the whole exhibition, and it died halfway. And the media obviously made a bit of a sensation out of it, but it also generated, from my perspective, a much more interesting conversation, because then people were starting to talk about the ethics of it, the responsibilities that we have towards life forms, created a much more nuance rather than, yay, we're going to save the world. And this is, again, the difference between art and design, because my designer friends came to me and said, you failed. You didn't manage to keep it alive for the whole exhibition, you created this whole story. But my artist friend would say you succeeded because you generated an important conversation rather than, you know, make it like smoothless, as if there's no issue around it. So, you want to talk about meat? Right, so before the victim study, we also grew a piece of meat. This is the very first piece of meat we've grown, actually, again in Harvard. So there were lambs. We were working with a scientist who was doing in utero tissue engineering, and she harvested what's called satellite cells. So those are the progenitor cells of muscles. And she had so many of those that she didn't know what to do. And we said, okay, bring it to us and we'll grow meat. So this is the first piece of meat that actually grew really well. It looked quite fleshy, but we couldn't eat it because it was a biomedical research lab. And there's obviously lesson number one you learn when you go to a biolab is then never put anything in your mouth. Yes? So we couldn't do it. But then in 2003, we were invited to a show in France that had enough budget and to keep us there for the whole exhibition, but also for us to set up a fully functioning tissue engineering lab with brand new equipment, so there's no risk of cross-contamination. And we decided to grow our meat there, and it kind of made sense because France, obviously the culinary tradition there, food is really important for them. But we also knew that for French people, the idea of eating engineered food was very unappetizing. And for most other cultures, the idea of eating frogs was very unappetizing. So we decided the first piece of lab-grown meat to be consumed would be frog steaks, or in vitro frog steaks. Again, because it wasn't about trying to sell it as a solution, it was like how strange it is to think about the possibility that one day we'll be able to eat meat that was never in the body of an animal. Right? So if we are what we eat, what does it mean? What does it mean to have meat which wasn't a part of an animal? And the design of the lab was kind of corresponding with with the Alexis Carrel lab that I mentioned to you, and also with the architecture of the building. It was an old cookie factory. And then what was quite unique, it had a dining table. We also rescued 2 frogs from the animal frog distributor in town, so every day we would go and feed our steaks, and feed the frogs. And then at the very last day of the exhibition, we had this ultimate nouveau cuisine dinner. We grew about 5 grams of this meat, and we served it for dinner, and then we also released the frogs in the ponds of the botanical garden. So what the video I'm going to show you is, if not the first time anyone put into the mouth a piece of meat that was grown in the lab, because who knows what scientists are doing at night, but this is the first documented time where anyone put into the mouth a piece of meat that was grown in the lab, so you can see. The problem we had is that, here we are, and this is Joe Davis, a very famous artist. Looks so young there, huh? So, as you can see, it was kind of hard to cut because we were limited by the timeline of the exhibition, so the polymer didn't break down completely. We were using frog muscle cells, but we didn't exercise them, so they were a bit like jelly, so the texture wasn't that good. But because I was concerned about health and safety issues, I asked the chef that cooked it for us to cook it in garlic and honey sauce. Okay. It was really nice, like really good antibacterial agents. But then, I also can tell you that 3 out of the 6 people that joined us for the dinner couldn't swallow it. It was just too much for them, and we kept those bits and we showed them as part of the follow-up exhibition. And part of that is also to show that we don't have to swallow everything the technology gives us. But then exactly 10 years later, this guy, Mark Post, the guy at the back there, a Dutch scientist, revealed his first lab-grown burger, and everything changed. Suddenly it became the sexiest thing possible. So it took more than 10 years for science to pick up, and the way he revealed it was very much as a performance art piece. Yeah. He did it in a film, in a TV studio. He had a TV presenter. The whole thing was cooked. There's no way to scientifically validate what happened there. Yeah. And another thing which was quite interesting is that his main baker was Sergey Brin from Google. So it's kind of known as the Google burger. But after he did it, there's about up to $20 billion now is being invested in research and development of this new lab-grown meat thing. So after that, which was also from our perspective, so since 2013, so up to that stage as artists, we would kind of create prototypes, working prototypes of things that we would think are going to happen. And then since then, to a large extent, we started to respond to what's already happening and how companies are, which kind of created a bit of a shift in our work. So here's just some examples of the follow-up. So here you can see the follow-up show, which was this 3-screen video installation on a dining table with the bits that were spat out by the people who couldn't swallow it, the victim cider that I mentioned before. And this is, in 2008, I was commissioned by BBC to grow another piece of meat, and I took it to Huston Blumenthal, which is the very famous British chef, for a TV show which was around extreme technological solutions to global warming. And it was kind of interesting. It was like partly critical. It was okay, technology caused all of those problems. What happens if we throw more technology to solve the problems technology created in the first place? So I felt that was kind of an interesting approach. And also, actually, just before Mark Post, so a year before he revealed his takes, he had a press conference where he said he's going to do it, and I was able to invite him for a cooking show where I got him to cook against a philosopher to discuss the future of food, future of meat production. And I did it like a master chef style cooking show. And the secret ingredient was fetal calf serum. So it's the blood plasma of an unborn cow that is being used as part of growing cells in culture, especially muscle cells. So it was kind of an interesting thing. And I have so much respect for him that he basically agreed to do it. When I met him a couple of years later, he said that the Google PR people were giving him so much shit for working with an artist and that he'll never work with me again, but he did work with me on this project. And it became such a success that since then I ran about 8 or 9 of those cooking shows around the world, where I get scientists to cook against philosophers. Actually, the last one I've done in Australia, I got a scientist who's got a lab-grown meat company to cook against an indigenous farmer. And the idea was to look at, do we need to invest in technology or do we need to invest in traditional knowledge where we can find this future. I did also a piece where we made a soup, an insect soup, so I have a fly in my soup, and we literally, and the idea was that the nutrient solution and the insect cells would then be mixed together and would be served as a soup. And the way it was presented was what I refer to the dark cloud of resources, because most of the conversation about lab-grown meat are presenting the laboratory as a magical place where it doesn't take any resources. So for me it was really important to show actually there's a lot of resources that are needed in order to grow those things. So there's this big bag with the nutrients, that was kind of dripping it into this bioreactor there. Unfortunately, we got a contamination, it became a mushroom soup, and we couldn't serve it, so that never happened. And then also playing on the idea of many of those companies are talking about bringing the farm into the lab, and we decided to bring the lab into the farm and started a series of compost incubators. So the idea there was to use the heat that is being generated by compost to grow cells. And the one there on the right is the most successful one we've done. That was in London in winter, outdoors. So at night it went to -4. And it was what I refer to as pre-cybernetic. We didn't even use the thermostat, I just regulated the flow. So the way it works is that you basically coil tubing in the compost. The core of the compost tend to maintain a very constant temperature of about between 40 and 60 degrees Celsius. And then the hot water goes into a heat-jacketed incubator and keeps the cells alive. So this is kind of a different version of it. And we even kind of showed that the cells were not that happy, but we managed to keep cells alive for quite some time outside in London in winter using this technology. And I think we're getting better at that. The only place in the world where I was able to set up this installation inside a gallery, can you imagine like having 4 tons of compost inside the gallery, was obviously here in China, and that was in Shanghai. A very nice system, and obviously then it's much easier to regulate the temperature because you're indoors. But again, we didn't invent anything, because there is a bird in Australia that does this thing. So they, instead of sitting on the eggs, they invest so much energy in building those huge compost mounds where they lay their eggs, and they keep regulating the temperature of the eggs inside this compost heap. And then just before the chick hatches, the parents disappear. And it's such a crazy story, because it takes about 20 minutes for the chick to get out once it breaks the egg and kind of goes out, and then gets out and takes its first breath. So for 20 minutes after it hatches, it doesn't breathe, but just climbs up and kind of breaks out, takes a big breath, lies and rests for a while, and then flies off. So it flies directly after. So But somehow, as an evolutionary solution, those birds decided to spend all of their energy building this crazy compost mound rather than just sitting on the eggs. All right, science can be quite strange sometimes. So this is a paper that was published in 21 by some MIT engineers, where they were proposing the use of basically growing wood in the lab rather than in a tree. So using the same logic of lab-grown meat, but in this case to grow wood. In the paper itself, there's very little reference to how the nutrients and the resources are needed, but the way they justify it is that, you know, if we just need wood, why do we need all of those useless things like bark, small twigs, roots, and leaves? Without mentioning the fact that the nutrients they need in order to grow those, this wood, would very likely would come out of an organism that would have leaves and roots. So this kind of strange logic that not everything which is lab grown is necessarily efficient or good, but seems to be generating a lot. It creates those very seductive stories, and that was at a time where money was cheap and venture capitalists would just throw money at whoever comes up with a really nice story, not necessarily scientifically. And that's what we started to call metabolic rift technologies. So the concept of metabolic rift is the idea that came out of Liebig, who was one of the very first scientists to develop artificial fertilizer, and that was adapted by Marx, where there was a realization that as human society moved out of the country and into cities, we literally stopped shitting where we're growing our food. So we would export nutrients from the country, but we wouldn't bring nutrients back. And the only way to compensate for that was to create artificial nutrients in order to feed the soil again. And so this kind of breakout as humans became more urban and technologically based, and it created this separation from nature and the metabolic cycles and forces that kept us with nature. And in a sense, all of those technologies of trying to grow lab-grown stuff are almost celebrating this idea of the metabolic rift. And so we did a series of works around it. This is the latest work that we've done in Waikam in Japan, where we created this food cycle. And the idea is, again, using a compost incubator and growing plants. So what we've done was to grow meat, basically cells in an incubator that was heated by compost, but use the meat not to eat it, but to put it in an alkaline hydrolysis system, which basically converts it into fertilizer. The fertilizer is then used to grow plants in a hydroponic system, and then the plants that are grown in the hydroponic systems are then thrown back into the compost. So there's no food that is being produced, but there's a lot of data that is being collected that is then being kind of projected. Actually, I don't have the... And there's kind of a work with scientists, with an artist who's doing data visualization, so there's kind of, the data is being presented in such a way that no one can make sense of it. So it's basically a way to create huge amount of data, and it's kind of based on this idea that if the only thing that the cloud is going to rain on us is data, we're all going to starve, because obviously we're biologically based, not data-based entities. And I'll just finish with that. So we live, as I said, in a really interesting time. So on one hand, we are giving technology, human-made technology, agency and control to such an extent that they're becoming like sentient machines. At the very same time, we remove sentiency from biological beings, and I can prove it that this is Isha Datta. She's running a company called New Harvest, which is kind of a non-profit that is promoting cellular agriculture and lab-grown food. And here she talks about, rather than raise a chicken, in order to eat chicken meat, why do we need all of this wasteful stuff, so similar to the leaves and the roots. And here it's like, she puts those wasteful things like beaks, feather, and sentiency. And I think we really, as a society, have to have a conversation about who gives us a license to remove sentiency for other living beings. just to satisfy our own pleasures or needs. Yeah. So what is our place within the living world is the question which is an extremely important one at a time where life becomes a technology and biology becomes engineering. That's where I am. So, thank you.
Cool. Thank you. I think so. Obviously, biotechnology can remove sentiency from other entities, but at the same time, it can also create new sentiencies. That's the stuff we working with, like cell, right?
Exactly.
We're starting with non-living things,
Exactly.
just organic molecules. We want to create a sentiency that non-exists before.
Exactly. No, this is really interesting you're saying it, because last year I organized a conference called Synthetic Sentiency, which was exactly about kind of the new ethics and politics of new forms of sentiency, either AI or new biological sentiency. So things like brain organoids, but even what depends on what level of complexity you want to insert sentiency. But you're totally right, and that's the really interesting with this thing with the synthetic cell. Many years ago, there was a Swiss group that was trying to create a synthetic cell, and they ended up employing a philosopher. Because they said that they would only know if they're successful if the philosopher would say that they made new life, because life is a philosophical, not a scientific question.
And we invited a philosopher 2 months ago to visit us, right?
That's right.
And his comment is like, we won't be able to do that within 10 years. So,
Do you know Hideo Iwasaki, synthetic biologist from Japan? One of them, he's running an art and science. Actually, you should really connect with him.
Yes, definitely.
At Waseda University. I think quite a few of us. You're working in them.
He's professor.
Okay, great.
But he's also a philosopher, and he works a lot with philosophers because he's really curious about those questions.
Yes.
I think and obviously there is Western approach to life, and then the Eastern approaches to life, and I think we need to find something that.
Well, at least in the first place, bring people together. To know what each of us stands and arguments.
And I think also, even in a very practical way, this is an important strategy to prevent too much pushback.
Yes.
And you want to be able to engage in those conversations before literally the shit hits the fan. So you want to be there. You want to show that you're engaging in those conversations, and there's a solid reasoning for how those things are going to be developed and deployed.
Exactly. That's cool. Thank you.
Better.
We can open to a discussion and Q&A.
So I don't know.
Actually, I'm from AIRS, which stands for AI and Robotics.
Okay. All right.
So I think this is a brilliant idea because, I was sharing. I was keep thinking about how to view the concept of life in today's world. Because would you think artificial intelligence is also a form of existence of life, or do you think it's more related to biostructure, biosystem, and how many
So for many years, I would say I'm carbon biased. So I'm a carbonist. Silicon, I'm not interested. But lately, I'm more and more interested because I actually heard a really interesting podcast where someone was saying that the way we engage with AI, because we evolved to be a social being, and if AI is starting to respond to us as if it's living, as if it's conscious, as if it's sentient, and if we treat it badly, we will start to treat each other badly as well. So it's not even about the silicon, it's about how we as humans, as a culture, are treating other things that even if they're not fully sentient, our relationship to them would impact also the relationship we have with each other. And something I very important in my work is to not just talk about human life and be kind of non-anthropocentric, but with the idea that if history taught us anything, is what we choose to do to other life forms, in some stage we would start to do to ourselves. So when you think about eugenics, and eugenics came from the idea of scientifically breeding farm animals to be better for what we need. And actually the Center for Eugenics, the main place where eugenics started was the Cold Spring Harbor Laboratories in America, which was an agricultural research institute that then became a eugenics institute about improving the human race. And then the Nazis kind of perfected it in a sense by all of the programs of exterminating part of human society. So even if you're not constantly thinking about the human, you have to be aware that the effect of how we treat other things that are expressing qualities that we hardwired to think about as something related to, that would impact how we relate to other humans. So I'm now moving also to thinking about kind of the ethics of silicon-based entities, and because the question about life is a philosophical and not a scientific question, and there's no agreed definition about life, I would welcome silicon-based entities into my set of concerns, that's what, and considerations. Yeah.
I also feel like it might be another dimension of life forms or existence. Because ever since I was very little, I've been curious about like why the scientists go to the space, they look for aliens or life form, and then they want to find evidence of water. Maybe aliens, like their life logic is completely different from human or biological psychology.
Yeah, so there's claims that there's alien life, like not from another planet, but non-carbon based life already on our planet, but we can't recognize it because we don't. You know, they found bacteria in Antarctica that divides once every 500 years. Yeah. So it metabolizes so slowly that we don't even know, you know, it looks dead. So yeah, I agree that there might be other forms of intelligent self-replicating systems that we don't even recognize as being living.
Yes, maybe the concept of life can be expanded.
Right, so actually that goes, I gave lots of other talks around what I refer to as the poverty of language. So, you know, they're talking about the fact that the Inuit people, the people in the far north, have about 50 words to describe snow and ice. I know, I'm sorry to be a bit vulgar, but in the English language, there's also about 50 words to describe shit.
And in Chinese, actually, we have close to 20 words to describe fabric, silk.
Okay. How many words we have to describe all of the manifestation of life? How many words in Chinese you would have?
For describe life?
Life.
Maybe we should answer that.
I don't think many, a few. Yeah.
You know, the Greeks had two, zoe and bios.
Yeah.
We in English, we basically have one.
Yeah.
And it describes the whole manifestation of what life is. So I talk about this idea that we did, and this is, as an artist, I talk about the fact that I'm looking for things that we don't have words to describe. But because I'm not a philosopher and I'm not a linguist, all I can do is point a finger and say, we call this life, but this is different from this other thing that we also call life, and this is different from this thing that we also call life. We need to find new language. As you said, we need to find new ways to describe all of those things. Because, yeah, life, it's like trying to, and especially now, it's like trying to solve one of the most complex and kind of nuanced problem with a huge hammer. Yeah? And how you find, so this is exactly what we as artists and scientists to some extent, but mostly philosophers and linguists have to figure out how we as a culture find new ways to deal with it in a more complex way.
I'm also new to this institute. I don't know, I actually don't know how to introduce. I'm new friends to them. I just graduated from the Sydney University of Hong Kong last year, and I now teach at one of the local universities in Shenzhen. I was trained in art history, but for art theory, art history, that I focus on modern and contemporary art. And then for my PhD, I did a topic related to ecology and art, but specifically focused on the issue of indigeneity in the context of Asia, which is quite a niche field. And also I want to mention one interesting thing that we have this lab thing, but we don't call that lab. We have a group. In the group we have artists and theorists and art historians. We call that. So that group is called One Who Practice Group. It's a little bit borrow something from Taoist philosophy, but also are trying to apply that concept into how you wrap up everything happening in the world of ecological art, let's say. I'm overwhelmed by this lecture because I'm not expert in this topic specifically. I work more on the side of environmental humanities, so the philosophical debates between different or among different fields that are related to biology and ecology and art.
Yeah, obviously it's, and I just showed a very little part of, I've been doing this stuff for 30 years now, so there's many other things I could have shown, but I thought there'd be more scientists in the room.
We're working on it.
Yeah, fair enough. Are there any?
So I was thinking that
Yeah.
like, as an author who writes about contemporary art and tourism, I just have this feeling that people don't know how to, or we don't have we don't have certain standard or agreement upon how we should look at bio art.
Yeah.
Or what kind of aesthetic are needed or need to be established when we talk about those practices.
Yeah, it's really interesting you're saying it, though. As I said, I started with Yonazur, which is kind of artist-initiated residency in the science school, where we ended up building up Symbiotica. And then we were able to set up Symbiotica, and I could go two ways. Yeah. I could either say, this is my lab, I'm only doing it, no one else can do it, I found out a new way of doing art and I won't let anyone else come in. Yeah. Or what I chose to do is like, wow, this is amazing, I don't know, there's my approach, but I think there's many other approaches that need to be tested, and there's many other artists that should be exposed to it and see that, the very first, the research question, this is very different times. 30 years ago, I got funding with an open question, said, can living tissue be a valid form for artistic expression? Yeah. I didn't say this is going to be art. I said I want to explore if it's going to be, what it means. And I got funding, and for me it's still an open question, which is kind of interesting because obviously it's been going on for 30 years now, and for the new generation of artists, it's obvious that artists can use living tissue or biology. For me it's still a question, shouldn't it? It's like, so I'm really interested in seeing how other artists are approaching it, and that's why I opened up Symbiotica to those residencies, to set it up as an open place where it wasn't also me being the master and telling the people how to do it. I learned from them way more than they learned from me because they were coming up with other approaches and fresh ways. But I agree with you, there's still no definition. I have arguments, so I don't know if you know Eduardo Catt, and he's an artist. He claimed yeah, the Green Bunny, and he coined the term bio art back in 1997. And about 10 years ago, we decided to write a bio art manifesto, where the manifesto is like, a bio artist can only be a person who does this, 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 this. And he was trying to do exactly to standardize it, and he wanted me to sign it. And I said, no, I'm not going to put borders. I'm not going to say who is and what. And what's really interesting with Eduardo is that many of his works are, you don't know if he actually did them, or he tells us that he did them, but and actually I did some research about him, and I talked to scientists that he worked with. Some of his claims are not true, but this is another reading, and I have another talk where I talk about this idea that artists never signed that they have to tell the truth. Actually, artists are being celebrated for making things look like something they're not. So I can't, if a scientist comes up and say, I did this and this, and you find out it was fake, you get into trouble.
Of course.
If an artist does it, you might be celebrated, yeah, because, so.
Magic.
Yeah, and there's so many artists that call themselves bio-artists, and I know for sure that what they're claiming that they're doing is not real. And this is okay, they have a license to do it. For me, it was important for my own interest, because I'm interested in the materiality of things, to actually, and that's where the aesthetics of disappointment came. Yeah? I'm more than happy to disappoint my audience while knowing that I'm actually working with the materials and I'm honest. But I can't tell other artists that this is how they should do it. So, I refused, with Eduardo, I refused to sign his manifesto, but then I said, because I have a lab, I said, I'm more than happy to be a border police and see if an artist can fit your definition of bioartist. And I told him, let's start with your work. And he got so annoyed with me. Because he knows that I know that many of his works are fiction. Any more?
Yes. I'm so overwhelming and so admire and push it down for the last 3 decades. And I was participated in 2025 Ars Electronica Index.
Okay.
Yeah, I was collaborated with another artist. She was studying PhD at University in Linz.
Okay.
Concert.
Yeah.
And so I heard that there's a large place that year in Linz have like bio arts there. So I think that's starting from your work and the things other artists may follow.
Yeah, actually, Ars Electronica, lots of respect to them, because they started to get interested. Obviously, there were art and electronic arts, really, and the computer. I think it was 1995. They had a festival which was around life science, but there was no bio art there because there was no artist completely. But it was like speculating about it. Then in 1999, actually, they first showed bio art, and that was the work of Eduardo and another American artist called Gail White, who was doing work with mice. And then, in 2000, we set up the lab that I showed the Warriors was there. So they gave me the opportunity to show the first time live tissue engineered sculptures. Yeah. So they were the first serious kind of art organization that in 99, because Eduardo showed the genetically engineered bacteria, so the first time the genetically engineered organisms were presented as art. Even though there's an artist, Joe Davis, actually the one who ate the meat, he already did genetically engineered art in the 1980s, but no museum wanted to show it. So Ars Electronica was. So in this sense, I have a lot of respect to the fact that Ars Electronica, and then in 2007 they started a new category of the big prize, which was mainly around bio art. And again, I won't be humble, okay. I got the prize, the very, very first prize of this category back in 2007, without me even asking for it. And it was, they gave it not for a specific artwork, but they gave it to Symbiotica, to the concept of a platform that allowed artists to engage with biology as a symbol that this is kind of, it's not just the one specific artwork, but it is already kind of a place where this kind of stuff can happen.
After they establish it, I read an article and I found that it analyzed 41 years of art electronica, the themes, the change of themes, and the biggest one is bio arts. Yeah, it appears most in all the art electronica.
Since around 97.
Yeah.
Who get the second prize on that year? Never
Okay.
No one remember the second prize. Okay.
I wonder if they had this second
Let's
prize.
see.
Okay.
Okay.
I'm just curious.
No. See, I'm getting old.
Yeah, never mind.
Okay.
Yeah, and also your work is, I think there's a very famous book in our background. I'm from industrial design school, so there's a book written by the British.
Done already.
Yeah,
speculative everything.
Yeah. It's
Yeah,
very
yeah.
famous right now. It's like our school just launched the new curriculum and we just introduced that as a course. So one of our colleagues now is in St. Martin in London, the University of Art, and he studied PhD there, and his like mentor was studying that part of the subject. So it's a very honor for me that I come in after now years.
Yeah, so Tony and Fiona are really good friends of mine. Actually, I met them for the first time. They came to France for the show with the steak. And this is kind of interesting because they developed the whole concept of speculative design. I was designer and I decided I'm an artist. And they designers that decided that the kind of work that I'm doing is actually speculative design and not art. So we would constantly argue. They would call me a designer and I would be offended and I would call but then in 2015 they left the RCA and I replaced them. Now they're in persons. And I, for 2 years, but that was crazy. So I took their program, but I didn't want to live in the UK, I want to stay in Australia. So for 2 years, I would fly to London every month and run the program and then fly back to Australia for a month and run Symbiotica. But after 2 years, I was like, nah. and because Symbiotica was my baby, I preferred that and then moving to London.
So what do you think about those, I think before the pandemic, there's like huge fever about the man-made meat. And there's like a lot of capital, try to invest in that and they try to like promote it. But like after the pandemic, it seems like not that hot. And so what do you think about those? I think the idea is quite good and it's quite impressive. But like maybe in the future, like how it goes, what do you think?
Okay, so you know the Grafton hype cycle? Yeah. So the problem is that those companies promise too much. There's again, I have another lecture which I give only about that because there's a couple of things. It's not as easy. Or let's put it this way, I'm an artist, I'm not a scientist, I'm not an engineer. I was growing meat back in the year 2000, 26 years ago. I was able to do it. The issue is about upscaling and doing it cheap enough to compete with current modes of meat production. And ethics as well. But the idea is that maybe it would solve some ethical issues. The problem is that like in the dot-com kind of boom, the companies came out, got crazy amount of money, promised a lot, spent. There is a company called Upside Food that invested about $400 million in a big factory in Arizona somewhere, where they said that they're going to grow. Because the other issue is that you can't grow large chunks. You can only grow stuff that can be you can use for burgers, and you can so slurry, not really whole cuts like a steak. Because we don't have the capillary system, because when you grow cells in culture, they're not organizing themselves like real meat in a body organizes itself. But this company decided that they are going to be the first to have a whole cut piece. And they got the license, got FDA approval to sell it. They're working with this restaurant in Los Angeles, where they sell like those tiny kind of chicken meat pieces. And it's all about being environmentally friendly and all of those kind of things. In some stage, Bloomberg did an investigative journalism there to get some of the employees to tell them what really happens in the factory. So they couldn't get the machines to work. They ended up growing meat in one layer of cells in what's called roller bottles, and manually scrape it out and build it bit by bit, like one layer. So maybe 20 micron stick and build it into a 2 centimeter. So many. So I calculated for every piece of meat that they were selling, and they were selling it for like $5 to the factory, to the restaurant. They had to have something like 600 of those single-use water bottles, so not environmentally friendly. Labor was like crazy. They basically said every piece of meat cost around $10,000 and they sold it for $5. So they were basically burning their investors' money on this stuff. And then also the crazy thing is that it's easier to grow connective tissue than muscle tissue. So they were selling connective tissue, which is basically scar tissue, as if that's the meat. And once the story came out, so they were basically not giving the right meat, and crazily environmentally unfriendly, because those single-use bottles, you have to, first of all, before you can get rid of them, you have to sterilize them, and then you have to, so huge environmental costs. And that's not even talking about the food you feed the cells, yes, which also is really expensive. So once this story came out, it was like the story with Theranos, you know, the Elizabeth Holmes, you know that story? You know the,
My God.
Yeah.
Yeah.
Kind of very similar story of promising too much, not being able to deliver, basically cheating your customers and burning the money of your investors. Yeah, it's a typical Silicon Valley story. Yeah. And the other thing is obviously now money is not cheap anymore. There's not, you know, venture capital is very slick, and AI comes in, and it's like all of the money goes to AI. Yeah. So those companies are now falling. There's about 4 or 5 companies that just gone bust. It was another, I think, the third or fourth biggest company, an Israeli company, basically had something like $400 million in venture capital money. Burnt all of it. It was actually a lovely story that the employees were saying, yeah, they were investing money in crazy things because they don't know what to do with the money because the technology doesn't work. So like special security doors that cost like $10,000 a door, while the technology doesn't work, they had to find ways to spend the money of the investors. And so $400 million invested in them, and they now have something like $120 million in debt. So they basically spent $500 million and there's no product.
Wow, yeah.
So a couple of more. So obviously with the hype cycle, maybe at some stage it will come up again and people won't promise too much. I used to go to those, so they would invite me. I would come to those conferences, you know, those investors meet startup companies. And they would like bring me out and say, this guy, he did it way before us, but he doesn't know how to make money out of it. And shout at them and say like, what you're doing is stupid, you know. And my advice to them, because the way Silicon Valley operates is like to monopolize markets. Yeah, they don't, nothing is small scale. Yeah. So one company was like, we're going to be the sole supplier of meat to McDonald's. Another company was actually saying, we're going to do pet food out of it, which is like for lab-grown dog food, which is kind of ridiculous. The bottom line was that they would talk to those rich people and say, don't worry, it's not for you, it's for the poor people and for dogs. Yeah, but we're going to have a huge market. And I would say, actually, this is so stupid. This is like luxury food, and it's going to be luxury food. It's really expensive. Sell it as if it's like molecular gastronomy. But it's a tiny market, but you create a desire for it. And then but you're not going to get $100 million for that. You'll get maybe $5 million to start like a small company to do gourmet, specialized kind of novelty. No, we need to dominate the market. We want to be the biggest food company in the world. It's like, you do that, you fall, you know, So hopefully there'll be more of an adjustment, but I would say because the other thing is they're all quoting a scientific paper from 2011 of 2 environmental scientists that did a life cycle analysis, environmental kind of life cycle analysis of lab-grown meat, where they claim that it's going to use 95% less land, 97% less greenhouse gases, blah, blah, blah. You know, it looks very as if kind of magical, it's a magic place and I analyzed the paper. So for example, twice in the paper they say, you don't have to invest any money in incubators because the cells are producing enough heat and you would use that. It's like, this is not how it works. In order to create those hemostatic conditions, you need to invest energy. It's like you can't rely on the cells to do it. So I would go to those conferences and I would say, this paper is bullshit. And the authors, this Finnish girl, would always sit there and she was like, oh, yeah. And then when Bloomberg actually did this thing, they interviewed her, and that was like 15 years after she published the paper. And I have the quote from her where she said that it was a speculative paper, that she didn't know, and now she's much more pessimistic, and so she finally admits that it was wrong, but because the paper was never retracted, those companies still use it as kind of the sell pitch for it. Even though the author already says, no, that was a stupid paper, it was miscalculated. And so if you go to the websites of those companies, you would often see those kind of statistics about how, which is all based on this fraud paper. Yeah, because in a sense, what you're trying to do when you grow meat in the lab is you outsource the function of the body of the animal to technology. And everything relies, and all of the calculations will rely on the fact that all of the energy they're using is renewable energy. And it's the same now with data centers as well. So, we're not using energy because we're all using renewable energy. But this renewable energy could have been used for other things. Here in China, you keep on building coal power stations, and you're doing really well in renewable energy. But because the AI is sucking so much energy, everything, what's the name of this? There's a name of this thing that if you open more resources, people will take them. It's not that you're going to reduce consumption. And as the price goes down. So it's the same with that. And when you think, for example, the way they grow beef in Australia is they send the cows into the desert and the cow just eats plants that we humans cannot eat. And it's beside the fact that they're creating methane through their verbs, it's not such a big environmental cost. But in America, where they grow the beef in those industrial feedlots, where they use corn and soy where they feed the cows, which were never evolved to eat it, so you need to give them antibiotics as well. This is where the problem is. So when they say beef is environmentally unfriendly, if you grow the beef in the way the Americans are doing it, it is very unfriendly. If you just get them into the fields and get them to translate plant material that we as humans can't consume and make it into animal flesh that you can eat, that's actually quite an efficient way of doing it. And I think that meat is also a really, I refer to it as a symptom because it's not, there's a very simple solution to the problems of meat, and that's reduce our consumption. With humans, we never evolved to eat meat 3 times a day. It was always a special thing. So we would eat it once or twice a week if you were successful in hunting. But now, as many countries are becoming more affluent, there's an expectation that you would get your meat 3 times a day. But no one would invest in a company that is trying to reduce consumption.
Can I ask last questions? Like you explain more about the terms like the semi-living.
So it wasn't in this presentation. So I was talking about it last night. So when we started to work in the lab, we worked in a lab that was doing research into creating an artificial cornea for the eye. So they were trying to find, to see if the material, so it was a material scientist, but he had a tissue culture lab because he wanted to see if the cells from the eye are compatible with the materials that he was developing. And so twice a week the lab would get a cardboard box full of half rabbit heads. We were told the rabbits were killed for food in the morning. They would cut the head off and then send the heads to a brain research institute. and take the brains out, and our lab would get those half rabbit heads around lunchtime. We would then take the eyes out of the rabbits and put them in antibiotics for 24 hours and only start culturing the cells 24 hours later. So for us, not being trained as a biologist was like, we had a piece of dead meat. You can't be more dead than being a half rabbit head in a cardboard box. But more than 24 hours, and later we did experiments which we were able to isolate living cells from stuff that we would buy in the supermarket, from bones and stuff, from animals that were dead for 2 weeks. So we realized, and that goes back to what we talked about, the poverty of our language. Even though this thing is dead and can't be more dead than being a piece of bone in the butchers or half rabbit head, there's still life in it. So that's the life of the cells. So they're living, but they're not living in the same way as the rabbit lived before. So that's when we started to try and find a new language. And the idea of the semi-living was, but then we also made it, kind of added another layer, and that's, from our perspective, semi-living is life that is being supported by technology. So it's alive, but it cannot survive without technological support. Then we found out, actually, so the Tasmanian devils, those small, they have a kind of cancer which is contagious. And the reason for that is that one Tasmanian devil got this cancer, and they're very aggressive with each other, so when they meet, they kind of bite each other in the face, and they transmit. So the cancer is still, it's like the HeLa cells, it's the cells of the original Tasmanian devil that had the original cancer. So it's not the cancer it's not like cancer usually, it's your own cells kind of going crazy. And this is the cancer that started maybe 20 years ago by one individual and just being transmitted. So you can claim that this type of cancer is also semi-living, but doesn't need technology because it's got the bodies of the other Tasmanian devils as a host.
What do you put in the worry doll? So you said the worry dolls are semi-living, right?
Yeah.
I see tubes. So you put some creatures inside the tubes to make it semi-living.
No, we used, so we're growing animal cells in them. Actually, the very original worry dolls that we've done in our Electronica, which makes it even more complicated. The way we worked was, we didn't want to travel with living things from Australia, so we asked our Electronica to find us a lab that we can work with in Linz in Austria. And they found a virology lab in the hospital that was working with living cells because you can use as a way of diagnosing viruses. There's actually cells, you basically infect cells and then you can see what virus it is. So they had like they're working with about 10 different cell lines. So cell lines are, so they send us the list of all of the cells that you are using. And one in particular kind of caught my eye. Originally, so the name of scientists give names to those cells. The HeLa cells, it's because of Henrietta Lacks. There's the Chow cells, which are Chinese hamster ovary. But then the one that this lab was using was called McCoy. And I think it was named after the original scientist. But also McCoy is the name of the original doctor from Star Trek. So I said, I want this cell because this is because I'm a Trekkie. So I want McCoy cells. And then I did research into those cells. And apparently the original McCoy cells came from a knee fluid of an arthritis patient. And they made a cell line out of it. And it was distributed around the world, considered to be a human cell. And then at some stage, someone did the genetic testing and found that those are mouse cells. Because somewhere along the line it was contaminated with the L strain mouse, which was the original cell line from the 1940s. But it kept on being distributed as humans for many, many years. And the stories now about a lot of breast cancer research had to be pulled out because some student did research and found that the main breast cancer cell was not what they were saying. So, tissue culture world or cell culture world is really messy. So, which added another really interesting part of the story. Yeah? So suddenly it's not what you see. So we're using McCoy cells for the original worry dolls, but then when we showed it in other places, it depends on what cells are. Because in the context, with some of our projects, it's really important for us to know what the type of cell it is, and so we had a show in LA where Henry showed and Tucker were showing their work. We were very specific about using a cell line which is a mouse mammary gland cell, because it was standing for the production of lab-grown milk. So it was important for us that those are the specific cells that are growing in the gallery. But with the worry dolls, it didn't really matter for us what type of cells they are, as long as they are cells and they're growing over the polymer. But it's always mammalian cells, but from our perspective, it can be human or animal. In some cases, also there's no difference. And the McCoy cells kind of show this. Sometimes even the scientists don't know the difference.
Have you ever met Pat Brown? This scientist from I think Stanford. Met Pat Brown, and he's the guy who founded Impossible Food.
Okay.
Yeah, I think so. You're talking about like the macro needs and the market stuff, but as an alternative to meats, there's another totally different strategy, right? So let's just harvest soybean, because we drink soy milk, so they are very rich in protein, right? So the idea is to process the soybean protein so that it tastes like meat. Right. So that's rather than.
But when possible, also the genetically modified yeast produce the heme, which is like blood. So
So
the more blood,
it adds the special flavor of meats. Right.
The only problem with all of those, and I like them, when I'm not in China, I try not to eat meat. When I'm here, I eat everything. So in Australia, I would take my daughter and go. There's a burger place, because she loves burgers, and she will get a burger, and I'll get the Impossible Burger. But it's not healthy. It's highly processed food. So as a treat, it's okay, but I wouldn't eat an Impossible Burger every day.
So I think that that problem might be solvable by technology. So the reason I brought him about is, so I was listening to an interview on a podcast, and it happened to be an interview with him, and it was maybe 10+ years ago. So that was the most inspiring and enthusiastic interview I've ever listened to. So he had this remarkable story, of how he got into the field. He was extremely successful scientist. He was a member of the American Academy of Engineering. The big league. So he invented microarray, which is sequencing technology, which he can sequence a lot of things at the same time. But when he got his 50s, he felt, okay, I've done all my work in the past. Now I want to pick another problem that's big enough, and I'm within my reach. So he just picked an alternative to meats, because obviously, farming actually, farming the animals is the single biggest impactor human activities on the planet, right? So if you can get rid of that, then that save the planet literally. So then that's how he come up with the strategy. Okay, we can plant plants. So plant plants, no problem. Farming the crops and fruits, and then. Well, that's how we raise animals, right? We feed the corns, like soybeans, to cattle, to pigs, and then they produce meat for us, right? So there's a fundamental ecological principle behind that, right? So we know the food web concept, right? So we have what we call like the primary producer, which are the plants, because they basically use the free energy from the solar and convert CO2 into biomolecules, okay? And then animal eat the plants, and we eat the animal, right? So we are on the third level of the food chain, right? So the problem there is each time you go from one level to the next one, you waste the mass majority of the energy, right? So in other words, the food web is a very inefficient flow of energy. So that means if we can get our protein or food directly from the plants, so we basically save all the would be wasted energy if we eat animal, right? So that's a like hardwired ecological facts, right?
Let me just stop you here for a second, just to talk about the efficiency of lab-grown meat.
Sure.
Because that's exactly the problem.
Yeah.
That the meat, the nutrients you feed the cells that you grow as lab-grown meat.
Yeah.
This is like already the first, So I talk a lot about the fact that we can just drink the nutrients rather than give it to cells to make it into meat, if it's about efficiency.
Yeah.
So it's never about efficiency, and this is what I would tell at those conferences as well.
Mm-hmm.
It's about aesthetic decisions, and
Yeah.
impossible to some extent. It's the same kind of thing.
Yeah.
We can eat soybeans. We don't need to convert it into something that looks like meat.
Yeah.
But we make an aesthetic decision that you want, and this is my decision as well. When I go and have the Impossible Burger, it's because I want to feel like I eating meat.
Well, that's true.
Yeah.
So if the whole, if everyone on the planet are Indians, then the problem solved.
Yeah.
Because most people in India, they are vegetarians, right?
Not anymore, as they're becoming more affluent. Now there's
Okay,
more-
yeah.
But there's a Buddhist tradition here. What's the fake meat thing?
Yeah, during vegetarian festival, people would
Yeah,
eat,
what is it called? Those fake chicken, fake.
It's a vegetarian anything.
Yeah, that's like over hundreds of years.
It's
Yeah,
basically
tofu or
cognac or
wheat gluten.
yeah.
Good.
Made by being
That's
Yeah.
That's
But
why
they
we
make it look as if it's meat.
Yeah, and so make it taste like meat.
Exactly. So you don't need a startup company. You just have to come to China and eat traditional.
That's why the market does not exist in China, because you already have that
Yeah,
all
exactly.
times. Yeah. Well, again, my point is, the point I wanted to raise is, what's the same we discussed earlier, right? So it's the technology versus human desire.
Yeah.
Right, so
Yes.
it's all the way down to
Yeah.
this problem. And we have a big dire backlog, which is a few years ago, the world population just exceeds 8 billion. Right? So that we have to do something, otherwise, the Malthusian, whatever tragedy will arrive.
So here's an interesting thing. They're saying that AI is going to take so many jobs.
Yeah.
And if we were smart as a society, we have to train all of those people who are losing their jobs to be small farmers.
Yeah.
And then,
100 years ago.
yeah, exactly. And people feel in COVID, I don't know if it's here, but in Australia, so many people started to have food gardens.
Yeah.
Because they were locked in and they were feeling really good about it, because, eating the food that you grow yourself makes you feel so much better about, So the government have to give subsidies for people to just go and become like small farmers and feed their communities.
Yeah.
And then everyone will be happy. And AI can do the boring stuff. Like science and coding.
I think it's also a very traditional, Oriental or Eastern kind of idea as well. Go back to the countryside.
Yeah, because in Japan, for example, the average age of the farmer now is 65. No young people want to do farming. And Japan has a very long tradition of small farms. It's not like the American style mega farms. But no one is doing it. They're dying out.
I actually want to know what's the flavor of the frog steak?
The flavor was hard to know because the sauce was really good, dominating the garlic and honey. But speaking about flavor, I was in one of those conferences, and one of the companies was like a special ceremony. They got actually Isha Data, the last slide, the woman from New Harvest, and me, and this company in a ceremony gave both of us the meat they were going to taste in front of the whole audience, like 500 people. And they made the meat like a dried meat, like a jerky. Yeah. And we eat it, and they asked us, what do you think about the flavor? And it wasn't me, she said, it tastes like dog food. And it did taste, because they were using flavors. You need to add flavors to those meat, because there's no blood, there's no fat. Yeah, so you have to, now they're trying to find ways to combine it. Many of those companies now are basically doing this combination of like impossible and human and animal cells. Mm-hmm. So you would have like 5% animal cells and the rest would be plant protein. And then flavors. So,
Mala.
Is it true?
yeah, it depends on, but this is like processing it like processed, like what they're doing with vegetarian meat. Yeah, so they have to, there's a lot of processing they have to do to create the texture and the flavor. So even though they're trying to sell it as if it's like any other meat, it's very, very processed.
Okay, cool. Okay, thank you so much. And then
Speaking of, don't have the money for the, but
lab grown tam-tam.
cherry. That's the Australian flavors of mint chocolate and
Yeah, so we'll have our happy hour, 6 minutes rest, and chill out, and we'll have happy hour.
Okay.
I think more signs will join us for drink and food.
I can't.
Yeah, you should try it. It's sweet, it's good. You like sweet stuff, no?
Yes. I mean,
That's from Australia. Those are very famous Australian cookies.
I just saw the message.
But it started, it was always very simple, just chocolate. Now they have all those crazy flavors. Yeah.
Well,
And then,
same place. Huh? What?
Actually, also got very inspired by the practices of their as we also are having like because
Very good. Really bad.
from the memorization to practice inspection. And also with your residents as well.
Thank you. Don't take a WeChat, but WeChat doesn't like me. Just trying to send me messages like to register on my phone and never got them. Yeah, that'd be great. So it's basically.
So every time with the residents, we will prepare a document called China Survival Document.
Actually, yeah, you need to hear that because I need a China survival document for this resident. Luckily, I got Henry.
Yeah.
Here's my China survival document.
Yeah.
I think if you want to get international.
Yeah, definitely. That's exactly where we invite you, right? So we just freshly open, right? So you totally know, yeah.
It's like a lot about VPN and treatment.
Exactly. It's so because it's so different from the West.
Yeah.
So yeah, a bit traditional. I give you. Yes, please.
So my institute is also supported by CHK, which is
Okay.
Chinese University of Hong Kong.
All right.
It was the AI and robotics lab in the university, and the wages were government
And do you, so you get international residents?
yeah, it's every year from January to February we bring 10 MIT graduates.
Oh, okay.
Yeah, it's already This year, this year is the second session, then certainly.
Yeah, because my university has a very small art school, but I'm trying to push now to get the art and technology for us bigger. And, yeah, we're on the west coast, so we're in the same time zone. So it's really easy. 8 hours flight from Hong Kong. So we're the closest from the big cities in Australia. We're actually the closest because Sydney and Melbourne, I think it's like 12 hours flight or something.
So south of China, so it's
Yeah, there's direct flights from Hong Kong to Perth.
how many days?
So 2 weeks. I arrived here on Saturday, and I'm flying not this Sunday, but in a week. So, My understanding is that if you're not criticizing the government, you're okay. And because I used to travel a lot to Shanghai and Beijing, so I went living in Kaffa, and I would do studio visit. I actually find Chinese art really interesting because they have to find more complex ways to express their ideas. And rather than, so it makes artists that have something to say, makes the way they're saying it, for me, more interesting. It's like a puzzle. Figure out what it really means. So I actually, but in the West now, in Australia, for example, we do have censorship as well, but differently. So that the council. Yeah, exactly. Political correctness, and now there's things about you can't say anything against Israel, for example. So I think that's, I don't know if it's good news or bad news, but in Western China, starting today, China is building up a bit, the West is slowing down, so it's getting worse.
I think they were looking So the reason which you said before was that we thought the movie would be very hot. But
Because I did work here,
let's try the second one first. Okay, yes.
Last year, March.